Good morning everyone, my name is Jedric and I am here with Xiang and Lucas. Today we will be presenting on human T-cell leukemia virus type 1, also known as HTLV1. HTLV1 is a retrovirus, which means it uses RNA as its genetic material. Once it infects the person, the virus converts its RNA into DNA. This viral DNA is then inserted into the DNA of the host cell. As a result, the infected cell can continue producing more viruses, allowing the infection to spread to other cells. One important thing about HTLV1 is that it causes a chronic lifelong infection in humans. For this presentation, we will focus on 5 main areas of the HTLV1 infection process. These are adsorption, signaling, penetration, uncoating, and targeting. These steps help explain how the virus attaches to the cells, enters them, and affects normal cell functions. First, adsorption, also known as attachment, is the stage where HTLV1 attaches to the host cell. The virus does this through its surface glycoprotein, which helps it bind the receptors on the host cell. This attachment is important because it is the first step needed for infection to begin. HTLV1 is also described as a cell-associated retrovirus, meaning it usually spreads through direct contact between infected and uninfected cells, rather than mainly floating freely as virus particles. After attachment, the virus can fuse with the cell membrane and enter the host cell cytoplasm. This process is supported by viral glycoproteins, including the transmembrane glycoprotein, which helps mediate fusion between the virus and the host cell. Next is signaling. HTLV1 mainly affects host cell signaling through a viral protein called Tax. Tax activates important pathways such as NF-kappa B, which helps infected T cells survive, multiply, and avoid normal growth control. Tax also increases viral gene expression and disrupts normal signaling pathways linked to cell activation and multiplication. Besides Tax, other viral proteins such as P8, P12, P30, P13, and HBC also help the virus persist in the body by affecting immune responses and cell survival. Overall, these signaling changes allow HTLV1 to manipulate the host cell and maintain long-term infection. Thank you very much for listening. I'm going to pass it to Lucas now.
Thank you, Jedric. Moving on to penetration. Penetration is a stage where the virus enters the host cell. For HTLV1, the main penetration method is membrane fusion. Membrane fusion occurs when the viral membrane joins with the host cell membrane, forming one continuous membrane. This allows the virus to deliver its contents into the cell. The process begins when the virus attaches to the surface of the host cell. This attachment causes viral proteins to change their shape. These changes bring the two membranes closer together until they fuse. Once fusion occurs, the viral core is released into the cell, allowing the infection process to continue. Looking at the steps of fusion in more detail, the virus first binds to receptors on the cell surface. This binding exposes a specialized fusion region on the virus. Next, the viral and cellular membranes join together, creating an opening or fusion pore. Through this pore, the viral core enters the host cell. This process is important because viral entry is the first step required for an infection. If membrane fusion is blocked, the virus cannot enter the cell and cannot replicate. For this reason, fusion is an important target for antiviral drugs and antibodies that help prevent infection. I'll now pass the time to Xiang.
Thank you, Lucas. Now I will be going through the targeting of the human T-cell leukemia virus. This virus mainly targets the CD4 T cells that serves an important role in signaling other immune cells to identify and attack pathogens. The virus uses surface unit glycoproteins to bind to target cells. Entry molecules of the target cells include HSPGs, NRP1, and GLUT1. These molecules help the virus bind, attach more strongly, and enter the host cell. And this is what glycoproteins are. After entry, the virus goes through internalization and uncoating. Uncoating means that the virus capsid opens and releases the viral genome and enzymes into the cell. This allows reverse transcription, where viral RNA is copied into DNA. If uncoating happens too early or too late, infection may fail. Uncoating is still an active research area, especially in understanding how virus stays stable before release. Thank you, and here are the references.